Hi, I'm Professor Johnson, and this is the first in a series of two videos where we're going to be talking about the major macromolecules in biology. In this video, we're going to be going over some of the basics and then delve into the two categories of carbohydrates and lipids in a little bit more detail, while we'll investigate nucleic acids and proteins in the next video. So something that's common with all the macromolecules are that they are organic. Now when I say organic, what I'm talking about is their chemical structure. In this case, it means that they contain carbon molecules. And carbon's going to actually act as the structural backbone for all of these molecules. Now what makes carbon great for this is that it contains 4 valence electrons, as you can see in this diagram here. So this allows carbon to bond in a variety of ways. For example, you can have just a straight chain of carbons, as you see here. You can actually throw in double bonds here. So you still have 4 bonds going to each of the carbon, but more electrons are shared between some of the carbons than others. You can also have, instead of just a straight chain, branching that is coming off from the carbons. Or you can get really complex structures such as a ring here. And the versatility of carbon is what really makes it so great. The different ways that it can bond allows for a lot of different structures, and so our biology can take advantage of this and use it in a variety of different for a variety of different important molecules. Now, what makes the different molecules different from each other isn't the carbons per se, but the different groups that come off from it. So you can have functional groups such as the methyl group shown over here, which is a carbon with 3 hydrogens coming off from it. You can have hydroxyl groups, so a carbon can be bonded to an OH group, which is known as a hydroxyl group. You can have a carbonyl group. So if the carbon is double bound to the oxygen in the middle of the molecule, you would have a carbonyl group. Now if you would take the carbonyl group and the hydroxyl group and throw them together, so you have double bonded O over here and you have the OH, you can have a carboxyl group. So you can see how this is the mixture of a hydroxyl and a carbonyl in its name as well as its structure. The carboxyl group and this next group, the amino group, are going to be important players when we get to the amino acids. So this is a nitrogen with some hydrogens coming off from it. You can also, instead of an oxygen, have a sulfur in here. So you can have this as a sulfhydryl group. This is also another important player in a specific amino acid, which we'll talk about in the next video. And then finally, you can have, well, not, and then another one that is important is the phosphate groups. So this is a phosphorus with 4 different oxygens coming off from it. Important in the nucleotides. It's an important modifier in proteins as well. So we'll see this in a variety of places throughout the macromolecules. Now, in order to form macromolecules, you have to take and put together different polymers, or different monomers, meaning the individual units together to make polymers. So in order to do this, you need to have a reaction that will bring these together, and you also need a reaction to break them apart, and that's what hydrolysis and dehydration reactions are about. So both hydrolysis and dehydration, you might expect contain water or in the equation, and they certainly do. So a hydrolysis group is going to take a water, hydro meaning water, lysis meaning break it. It's going to break the water and it's going to break the macromolecule in two. So you can break off the different monomers of a polymer with hydrolysis reactions. Dehydration reactions are also known as condensation reactions. They're the opposite. So you would take a water molecule out of the overall molecule and attach the units together. So dehydration reaction is bringing things together, and hydrolysis is breaking them apart. All right, so now let's take a look at the carbohydrates. So carbohydrates are going to have this main structural units that make them. So they have a carbon, two hydrogen, and an oxygen. And then you can have these in different units. So you can have just one of these, you can have 6 of these, as we'll see with glucose, and they're going to be brought together. Now, when we look at the monomers of carbohydrates, we're talking about the monosaccharides. Now, there are going to be some important ones for you to know. So glucose, fructose, and galactose are your 3 6-carbon monosaccharides. So these are all hexoses. They're going to be the ones that are most nutritionally important. They're going to provide the energy. Glucose is particularly important. It's our blood sugar, what makes that up. But the fructose and the galactose nutritionally are important as well. You can see these 2 pentose sugars, ribose and deoxyribose. These are going to be important to know, but we'll talk about them more when we get to the nucleic acids. And then there's glyceraldehyde. This is a 3-carbon one. This is going to be important when we get to glycolysis. Now, you may notice that we have 3 sugars that are all have the same composition. So they're all C6H12O6, and they all have 6 carbons. What makes them different? Well, what makes them different is how they're structurally put together. So you can see that with fructose, you actually only have a 5-carbon ring because you have one of the carbons coming off from the chain. With glucose and galactose, you're going to have a difference in how the actual hydroxyl groups are coming off from the glucose. So these are actually isomers of one another. They're structurally different even though they have the same chemical formula. And with glucose, you can actually have multiple kinds. So all of these sugars exist in equilibrium between an open chain form and the closed chain form. And when they open and close, how the hydroxyl group on the end ends up positioned can be one of 2 different ways. You can either have an alpha glucose or a beta glucose. And this is going to be important, especially when we get to the polysaccharides and look at how they're put together. Now, take 2 monosaccharides, put them together, you get disaccharides. And there's 3 important ones for this as well. There's lactose, which is the milk sugar. There's maltose, and then there's sucrose, also known as table sugar. So sucrose is made up of a glucose, that's this over here, and a fructose molecule. Maltose is made up of 2 different glucose molecules, and lactose is made up of a glucose molecule that's combined with a galactose molecule. Alright, so that brings us to the polysaccharides. So polysaccharides, you start taking many of these individual monomers and linking them together to form the long chains. The two of the polysaccharides that are important are the starches and the glycogen, and these are really the glucose storage forms in animals for glycogen and plants for starch. On here I'm depicting the structure of 2 of the starches. Now it is important how the glucose molecules actually get put together. So if you look at a glucose, we can number these. This carbon over here on the right is the 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. Now, with the starches, you're going to have all of the carbons being 1 to 4 linkages. And then you can have some branching off that I'll talk about in a sec. But if we were to look at this, all of these are 1,4 linkages. It allows you to form a long chain of glucose molecules. Now, with this depicts amylose. There's another type known as amylopectin, where you actually have some branching coming off from the 6 carbon to a 1 carbon. So if we look at this, we have our 6 carbon right here. It branches to a 1 carbon, and so you can have branching coming off from this. And that's similar to how it is with starch. So amylopectin and glycogen are structurally similar. Glycogen ends up having more branching than the amylopectin, but structurally they're pretty similar. And so if you were to zoom out on this, this is how it would look. What this in the middle here is actually a protein that's important for the actual initial building of the glycogen, but all of these units are long strings of alpha-1,4 linkages with some 1,6 branching coming off. Now, you can have more polysaccharides than just the starches. So here we're looking at a cellulose. Notice that you're having alpha-1,4, so remember how the two glucose molecules can be structurally, or a glucose molecule can be structurally either alpha or beta. If all of them are alpha, you end up with a starch molecule. With cellulose, you're actually having a beta glucose being put together. Now this is important because cellulose is a starch. That means that the enzymes that break this down in our bodies can't recognize these linkages, so they can't break down the starches, whereas they can recognize these and actually will break them down in our body. So this difference is important with how the molecules are going to be used within the plants and animals. So cellulose acts as a structural carbohydrate, which gives the cells support, especially in plants. All right, so this brings us to the lipids. What's going to unite them isn't really so much any distinct structural linkage so much as they are all hydrophobic. So they all contain a lot of non-polar bonds. So there's a lot of carbon-carbon bonds going on here, like there are with others. But there's also a lot of carbon-hydrogen bonds, which is another non-polar bond. Now, if we look at structures, so we have some important structures going here. We have the cholesterols and the other, or the cholesterol and the other sterols and steroids. So that you can see there's a lot of carbon-hydrogen bonds with just a single hydroxyl group coming off of this. This is nonpolar. Cholesterol is important as a precursor for signaling molecules. It acts as a structural component of membranes, and it is an important molecule that our body uses. We also have free fatty acids. So if you look at this, you can see there's a lot of carbon-carbon bonds with some hydrogens coming off. And eventually this ends in a carboxyl group, also known as a carboxylic acid group. So you end up with free fatty acids. You take three free fatty acid molecules together and combine it with a glycerol molecule, and you end up with a triglyceride. So this is going to be an important molecule as far as providing energy needs as well. Both of these provide energy needs for our bodies. And can act as, and the free fatty acid can act as a signaling molecule on top of that. Phospholipids are really the base component of membranes. So you take a triglyceride, you remove one of the fatty acids from it, and you add a phosphate. Sometimes there's other things coming off from it, but you end up with this molecule that has a non-polar end. That's what the tails are. All of these carbon-hydrogen, carbon-carbon bonds make it non-polar. The phosphate, on the other hand, is polar, so you end up with a amphipathic molecule, meaning that it has one region that is actually polar and attracted to water, and another one that's nonpolar and isn't attracted to water. So let's talk about the fatty acids briefly first. So you can have either saturated or unsaturated fatty acids. You can also have different lengths of fatty acids, so you can add carbons and make the tail longer. You can remove them and make it shorter. What makes a fatty acid either saturated or unsaturated are the number of hydrogens it contains. So if you look at this one, this contains all the hydrogens that it can, so it is considered a saturated fatty acid. Now if it's unsaturated, that means that it can have more hydrogens with it. And how that occurs is you have a double bond between some of the carbons. So if there's just a single one, it's a monounsaturated fatty acid. If there's multiple ones, it's a polyunsaturated fatty acid. And how this is formed can actually occur in two ways. You can have it cis, so you can notice that the two hydrogens here are sticking out on the same side. You can't rotate between the double bond, so it's stuck in this configuration. Or alternatively, if you have it so one of the hydrogens is pointing in the other direction from the other one, you can have a trans fatty acid. So the cis fatty acids are the cis unsaturated fatty acids are the ones that are considered to be healthier, while the trans fatty acids are mostly artificial and generally considered to be less healthy for you. Finally, let's look at how the phospholipids come together to actually form cell membranes. So if we look at a phospholipid, notice that you have the phosphate over here that makes it hydrophilic, water-loving, and you have the nonpolar tails, which are hydrophobic. Since they are hydrophobic, water is not going to want to interact with them, and water is going to prefer to interact with itself, so these end up getting pushed together, and you have them facing each other such that you have this interior that's made up of the nonpolar regions, and then the exterior of these membranes are out here interacting with the water molecules because they're polar. So you end up with a phospholipid bilayer membrane. So if you're going to take this and actually disperse it in the cell, what you would actually probably have form is a micelle. So all the nonpolar tails are interacting here on the interior of the molecule with the polar heads occurring on the outside. Now with cells, it's a little bit more organized, like this liposome, where you have this interior region that has the water interacting with it, and then these layers in here that are interacting because they're nonpolar and not interacting with the water. All right, that's it for this video. You can join me in the next video when we talk about proteins and nucleic acids, and I will see you in class.